The broadcast is now starting. All attendees are in listen-only mode.
Good afternoon. My name is Jane Foreman, and I will be the moderator of today's webinar, New Tools for Presumptive Drug Identification in the Field. Before we start, there are a few housekeeping items I'd like to cover. First, all participants are in mute for the duration of the webinar. We will have a formal question time at the end of the webinar, but if you have questions during the webinar, feel free to enter them in the questions module that's on the dashboard of your webinar. If you have any comments during the webinar or want to stop us real quick for a clarification, please do that through the chat feature that's on the dashboard of your webinar. Finally, this webinar will be recorded and we'll send it out to all participants who registered for the webinar. You'll get a follow-up email from us in a few days. Today's speaker, I'd like to introduce Dr. Presumptive. He is CEO and co-founder of Centis. He has been instrumental in creating multiple products from the company's Ramen Spectroscopy and coated aperture spectroscopy technologies. Dr. Podoluri obtained his PhD from Duke University. Before we start, we do have a poll that we'd like people to answer. It's 2 questions, and I'm going to launch the first question right now and give you about 10 seconds to answer it. The question is, what method do you use to presumptively identify illicit drugs in the field? And if you could just quickly check, do you use color tests? Do you send it off to the crime lab? Do you use Ramen Spectroscopy? Do you use other, or do you currently not use any technologies? And I do see that people are answering. For this one, it seems like the majority of people are using color test kits, with a few people sending it off to the crime lab and a few people currently not using any test at all. The next question I would like to ask is, what method do you use to presumptively identify prescription drugs in the field? And if you can enter your answers here, do you use visual identification kits or go to the poison control? Do you use color test kits? Do you use Ramen spectroscopy? Maybe another method or no current method? Currently, I'm looking at the answers right now. It seems the majority of you are using visual identification or the poison control, with a few of you using the color test kits and a few of you using nothing in the field. So at this point, I'd like to turn over to Prasant, and he will deliver the webinar from here. Again, if you have any questions, feel free to pop up in the question toolbar or send us a chat. Thank you.
Thanks, Jane. Good afternoon, everyone. Thanks for coming and joining us for this webinar this afternoon. I'm going to talk about some of these new tools that are coming up for presumptive drug identity in the field. Please feel free to let the moderator know if you can't hear me okay, then I'll try to increase my volume here. So my title, or I guess my outline for the webinar is, let me change the slide here, is as follows. I'm going to talk about just the challenge of identifying drugs in the field, both narcotics and prescription drugs. Then we'll talk about some review of some of the existing methodologies that are out there for identifying drugs up there. Then we'll get into just Raman spectroscopy. I'll give you a brief introduction of Raman spectroscopy, and I'll explain how Raman can be used for analyzing drugs, and we can get into some examples of Raman spectroscopy, and finally I'll talk about our vision for new systems approach for identifying drugs in the field and getting into the drug introduction process. The main challenge is, today's law enforcement professionals are facing a growing number of drugs that they have to identify. Back in the days, it was just, your handful of drugs that are heavily abused, like cocaine, meth, marijuana, and even heroin. I guess those are the big 4. But now that world has changed significantly. There are several prescription drugs that are being abused. And there are bath salts, there are synthetic drugs, and even the regular, illicit drugs are also there. So it's just the universe of drugs that have to be identified in the field has grown significantly. The picture on the right that you're seeing there is actually a picture of a drug bust that has happened in one of our agencies, and you can see the number of pills that they're trying to have to go through and identify them is just basically enormous. And the biggest difference that has happened there is most of them are all drugs, and none of them are actually cocaine and meth that are traditionally being abused. So that's the challenge that the law enforcement is focusing on, is facing right now, but there are not enough tools to go and do any identification for them. And additional challenges are as follows. Again, like I said, there is an overwhelming array of substances that leads to confusion. The presumptive evidence collection is basically adding a burden for both the field officer and crime lab, because now the crime lab is also getting involved, even at the presumptive identification level. Previously, crime labs were only restricted for confirmatory analysis. Because of this growing number of drugs that are out there, even crime labs have to get involved to identify drugs at the presumptive level. And that's basically putting a burden on the whole judicial system, because you really don't know what to do with the suspects at that time. And most often the field officers have to release suspects, even though they know that they have a controlled substance, but they don't really have any specific evidence to detain them. And the crime labs basically receive several evidence, including uncontrolled substances as well, and then that just adds to their burden of workloads, and the average turnaround times is increasing significantly. So there are some new tools that are coming about to address that. And before I get into that, I want to at least point out some of the current tools that are being used. From a field officer's perspective, there are two options for them. Primarily, I think that is basically resonated well with the poll as well at the beginning of the webinar. One is the color kits. There are different brands out there: NIC kit, NAR kits. All of them work on the same principle. It's a fairly old methodology. They're low cost, but they're subject to considerable variance in use and interpretation. Plus, you have to carry multiple kits. And most importantly, they're only good for the top 4 or 5 drugs that you're usually dealing with, but it's not really good for the entire gamut of new drugs that are coming into the market. The other alternative for prescription drugs is strictly visual markings. You got your pill book, your drugs.com website, or poison control. Any of these sources, they all rely on what's the tablet color, and what's the shape, what are the markings on them, and then you usually identify the tablets based on them. It does not really get into any chemical analysis at all. And the forensic scientists at the lab, they use mass spectrometers. I think that's probably predominantly the common way of doing them. The analysis is fairly expensive because it involves quite a bit of time to go and prep the sample and then do the analysis. It's definitely favored and quote accepted because it's a very rigorous methodology. But the challenge is, you basically even have to send Tylenol samples to go through that process to rule out whether it is just a Tylenol or whether it's an abused drug at this point. Let's talk about traditional color kits. They are good for identifying a few select drugs, and they actually do a pretty good job in identifying them. There are multiple steps and multiple test vials that are required for identification. For example, if you have a white powder and you suspect that it's cocaine, and then you have a process that you have to follow through, and you get the cocaine specific kit, and you run that step, and based on the color of it, you tell whether it is cocaine or not. That's basically where the subjectivity comes in. You don't really know whether it's pink or light pink, and you're always guessing at the exact color of it. And the other thing is your evidence is actually destroyed during analysis, and these consumables are plus these consumables are perishable, and they must be restocked all the time. I think the average shelf life for them is about 6 years, 6 months, a year at best. Most important thing is these results are not reproducible because the sample is already destroyed. So you would have to get another fresh sample to go and do this analysis. They do a good job in trying to identify these drugs, but they do have some high false positive rate as well. What I mean by false positive rate is, they can turn the color pink. Let's say that the kit is for cocaine, and the color of the kit can turn pink even for other substances other than cocaine itself. There is published data on that as to in terms of what kind of precursors and cutting agents will also yield positive on these narcotic substances. So there's a high false positive rate as well, that these substances can go into, it can trigger the alarm when they are not actually suspect substances. So those are some challenges on color kits, and most importantly, they are limited to those drugs, and you can't really do any synthetic drugs or even bath salts to an extent with these substances. On the pharmaceutical end, on the pharmaceutical tablet end, the only way of identifying them is the visual methodology. That's the drugs.com, poison control, or even just going to a local pharmacy and having the pharmacist verify them. And that this predominantly it's poison control and local pharmacies verification is the main mode of operation for identifying these drugs. The methodology is the same for all of them. It's just following the pill book, looking at the picture of the pill, and trying to identify them. The problem there is, it's not a chemical identification, so you're not really, if the markings are removed, you can't really identify them, or if the tablet is crushed, it's not, you're not able to identify them. And even some jurisdictions, not all jurisdictions, allow the use of visual markings as presumptive identification as well. So these poison control times can be longer when you basically have to go and identify them. And these online searches can be inconclusive as well. So basically, what Ramen Spectroscopy does, it's a new tool that is becoming available for law enforcement to address these various system process issues. It sort of resolves that conflict in the lab professional who wants good science to do the analysis, and not really want the field officer to do exactly sample prep and doing analysis in the field. And that's basically where Ramen spectroscopy comes in, because it's an undeniable record of the substance if the interpretation is done correctly. And even if the substance is limited to specific drugs, like cocaine itself, you can yield significant productivity gains. So the overall system is just sort of strained by this workload that can be reduced by the use of these two new tools called, such as Ramen spectroscopy. So let's get into what Ramen spectroscopy is. And I know I'm talking to to law enforcement officers here, so I don't want to get into too much detail about the chemistry behind or the science behind Ramen spectroscopy. But I do want to give you an appreciation for how this device works, or how Ramen spectroscopy works. It's a vibrational technique like infrared spectroscopy that is being used for quite some time. Ramen spectroscopy is also an age-old technique. It's been invented, I think, discovered in 1940, I believe. So it's been there for quite some time. It's only becoming popular in the last decade or so with the advent of new lasers and new CCD cameras. It's becoming much more popular. The principle of operation is there is a laser involved in Raman spectroscopy that shines the sample. Most of the light from that sample is pretty much reflected back, and the key for Raman spectrometers is to basically block the light that is reflected back. And there is a tiny fraction of the light that is basically emitted in different frequencies that is representative of the chemical structure of the sample. So, and when you measure those frequencies, it shows up as a graph like this, where you have different peaks to indicate different bonds within the chemical structure. This information is very useful because this pretty much becomes a spectral fingerprint for the chemical substance that's basically being shined by the laser. So what Raman spectroscopy can be used then is to capture these fingerprints into a library and use those fingerprints to do the identification. Here is one example, actually 4 examples. The blue is the fingerprint for amphetamine. The red is the fingerprint for cocaine. The green is for ephedrine, and the last one is for methamphetamine. The only thing that I want to point out here is if you can see the location of these peaks, the height of these peaks, and where the peaks happen, they are all different for different substances. So cocaine would have a completely distinct spectral fingerprint from methamphetamine, and methamphetamine would have a completely different spectral fingerprint from ephedrine. So if a device or if a spectrometer can capture all these fingerprints and a library can be created, then identifying an unknown substance is very easy, because you would then place the unknown substance, you would get a fingerprint for that substance, and then you would match against the library, just like a fingerprint database, matching database would work in a crime scene facility. And once the match is made, and then the device can come back and say this drug is cocaine or this drug is methamphetamine. The good thing about Ramen spectroscopy here is this fingerprint is very, very specific to cocaine, so there will be no other substance that will have the same fingerprint as this one. So the chances of having any false positives is very, very low. It's a fairly accurate technology if used properly. So Ramen spectroscopy, what is its role in law enforcement? This is not a new science. It's actually, like I said, it's been discovered fairly 50, 60 years old. So it's a known technology. Most crime labs are familiar with it, although the most common ones that crime labs are using is mass spectroscopy and infrared spectroscopy. There is a worldwide body called the Scientific Working Group for Drug Analysis, and these are the guys. SWG Drug is the short name for them. It's actually formed by the Office of the National Drug Control Policy from the White House, and in collaboration with DEA. And these guys are tasked with coming up with some recommendations for the laboratories on how the seized substances and seized drug substances can be analyzed. So they basically looked at all the different sciences that are out there that are being used for drug analysis, and they categorized them into 3 categories: Category A, Category B, and Category C. Category A are the most powerful sciences, if you will. These are the ones that have fairly distinctive ways of identifying drugs. They are the highest discriminatory power is what they have, and then these are the ones that have very, very low false positive rates. Category B is sort of moderate, and Category C is sort of the worst case, basically. So if you look at all the different sciences, Ramen is a Category A technique in conjunction with infrared and mass spec and NMR and X-ray. So it's an accepted science as a kind of Category A technique. Color tests, on the other hand, they're Category C. Pharmaceutical identifiers are somewhere in between. They're sort of the Category B technique at this point. So the recommendations in general for labs, this is mainly meant for forensic scientists that are out there, that whenever they do an analysis for a drug, they have to do 2 tests on it. Usually it's a color test as a starting point, as a Category C, but the other test always has to be a Category A test. And most labs, since they're equipped with mass spectrometers, they usually rely on mass spectrometers as the process for doing the confirmatory analysis. But the reason I'm bringing this up is it just tells you the power of the science, that the Ramen spectroscopy science specifically, as to how it can be a good confirmatory science, basically. These Ramen spectrometers, again, they are not new devices. They've been coming out for the last decade or so. Here are some example devices that have been mainly useful for first responders. And the DHS community is very familiar with Ramen spectrometers, and they have been in use for identifying hazardous materials, explosive substances primarily. Most of them are fairly expensive. I think one of the most popular ones that is out there for explosive detection is $60,000 or even higher devices than that. At Centis, we basically have worked on Ramen spectroscopy mainly to try to basically bring down the cost of Ramen spectrometers and make it more affordable for law enforcement offices, local and state and local law enforcement agencies as well. And one such example is MFL 3000. This is basically a Raman spectrometer. It's just that we have the library of all the narcotic substances all packaged into one device, and we made it easy to use for law enforcement to go and identify substances. I just have 1 or 2 slides on MFL, and I'll come back to Ramen Spectroscopy in a minute. This is a Pelican-based device. This box that you're seeing in black is the Ramen spectrometer, and there is a computer here for doing the analysis to identify these substances. It's good for a wide range of substances, and like I said, this is the way where some system-wide operational efficiencies can be created by the use of these devices such as this. It's fairly easy to use, even though the science behind it can get a little complicated and the analysis of the spectra can get complicated. But if it is designed well, like MFL 3000, you can make it as a simple, easy-to-use device where you just basically place the sample, you press a button on the software, and all the analysis of the spectra happens in the background, and the law enforcement agency is just presented with an answer. This is cocaine, and we are this much sure that this is cocaine substance, and the officer can go back and take the action based on it. It's like I said, it's non-destructive, and you can use the same methodology for multiple drug substances. It's like one process for all of them, whether it is cocaine, meth, or bath salts. The process doesn't really change. You just place the sample here, press a button, and you get the answer. Some of the things that MFL 3000 can do, like it's a Raman spectroscopy-based device. It can identify narcotic substances, precursors, cutting agents, pharmaceutical drugs, street narcotic mixtures, and even synthetic drugs like bath salts. The library is fairly extensive, more than 3,600 different kinds of substances. Again, the power of Raman spectroscopy is as good as the library itself. So if the library is very limited, it is not going to do the job for you. So it's always important to have a very robust library, and MFL 3000 comes with a pretty robust library of 3,600 plus substances. There are no consumables in this, like any other Raman spectrometer, and there's usually not much factory calibration that's required. Most importantly, all the analyses are all recorded in the device, so you can actually create some simple user-friendly reports that you can present to your DA for showing the evidence, the presumptive evidence that you've collected in the field. It's battery operated. I think it lasts at least, I think it's rated for at least 6 hours, but I've seen it gone up to 10 hours or so. And it's a cop-friendly device is what I would call it. Let's look into some examples on how Raman spectroscopy can be used to identify several different substances. So I'll try to point out here 4 different examples to talk about each different case. So let's start with the drugs. The controlled prescription drug identification system. Like this is an example of an oxycodone, and this is actually a live example in the street where there was a pill. In fact, actually the pill was the markings have been removed on it. If the markings were there, you could have at least said based on the markings that it is oxycodone, but there were no markings on this pill. They have been purposefully removed by the crooks here. And the device, when you place the sample on this Raman spectrometer, the red is these lines, like I said, is the fingerprint for the drug. In this case, the blue was the measurement of this unknown tablet that was presented to the spectrometer. And the red was the library for oxycodone, and all the peaks were pretty much matched up, and we were able to identify that this substance was oxycodone. And we usually, from the user's perspective, they don't really see all this stuff on the left. All we want them to see is the answer. In this case, the answer is oxycodone. And we also give a stock picture of what oxycodone substance would look like. In this case, we showed the imprints of oxycodone are and things like that, so that if there are markings available, you can double check the results of this, and you have one other way of verifying what the substance is. Here is another example. This is an example of a cocaine sample that we measured in the field. This is again showing the spectral fingerprints of what we measured. That's in blue. And the red is what the unknown substance, the reference substance for cocaine is. And as you can see, most of the peaks pretty much matched up, and our algorithms were able to identify that the unknown substance is actually matching with cocaine, and then they were able to give it the answer, and the device would come back and give a simple answer. So in this case, the answer would look something like it's cocaine HCl, it's it's 100%, and the officer can go back and take action by filling in some title, case information, and things like that, and then getting in there. Again, the science is pretty powerful, but we also want to make it simple enough for the user to not get into the science on how analysis works, but just get an answer for what the substance is. So what about street samples? Cocaine is never available in the street as cocaine. It's always cut with something like caffeine, baking soda, things like that. Here is one example. Actually, I want to show you a street meth sample. This is actually cooked meth, and dimethyl sulfone is one of the cutting agents or one of the remnants of the meth that actually happens. So whenever you have meth, there is also some remnants of dimethyl sulfone that is available in there. So in this case, what happens is if you look at the measurement, which is the blue, and meth sample, meth spectra, which is on the red, you see a bunch of extra lines there that don't really add up. You see a lot of overlap with the meth samples, like these two peaks are matched up, these two peaks are matched up, few peaks are here are matched up. But there are these big peaks that are not basically being matching up with what's in the meth spectra. And that's basically the other substance, but the device actually, if the algorithms are designed well, then you can pretty much go and do that analysis in the background itself to see if there are any other cutting agents that are mixed in there. So in this case, we were able to find out that there is a dimethyl sulfone, which happens to be a pretty common cutting agent, has those peaks that were missing. So the blue is the measurement again, the green is methamphetamine, and the red is dimethyl sulfone. So wherever there were peaks that were not explainable in the meth sample, they were all there in dimethyl sulfone. So if I had combined these two together, then all the peaks would have been exactly the same as what my measurement is, and this is basically what we are showing. So the device, from the analysis perspective, will just go back and does the analysis and say, I see two things in the substance. It's methamphetamine, and then it's also dimethyl sulfone. And the device is also good enough to go and give you some relative metric on how much meth is or how much dimethyl sulfone is there. Again, like I said, these numbers our percentages are mainly meant to give a relative understanding of if you have two different samples of meth and dimethyl sulfone, you can use these relative ratios to identify which one is more concentrated in meth and which one is not. So sometimes you can use that information to see if you are closer to the source or if you are basically farther away from a source. The example would be if you have a pure cocaine, that means you're closer to the source. If you have cocaine is cut with caffeine and you're seeing very diluted cocaine coming out at the end of it, you're basically further away from the source. And you can perhaps use this information this is pretty much a forensic information at this point. You can use this for your intelligence gathering at this point. NIC kits usually don't give those kinds of details. NIC kits just basically tell you it's cocaine. They won't give you whether it's a pure sample or if it's a much impure sample where there is very little cocaine present in it. That's the power of Ramen spectroscopy. Again, is you can get that information there. Now, I'll give you one other example. Bath salts are becoming a big issue that is out there. There is actually methylone or metcathinone. I think it's DEA has added this substance to the Controlled Substances Act. I think it's a C1 drug now. I think it's a recent phenomena. It's been added to the DEA list in the last couple of months or so. This device, Ramen spectroscopy in general, can be used to identify those bad salts as well. They would exactly work as the same way as cathinones. So here is one example where we have a measurement and it matched with metcathinone. Again, there are a few other substances that are from the cutting agent, and when we did the identification, we were able to find out that there was one other substance that's present in it. I think I'm cutting that measurement, the other substance in here. I think it was methylone in there. Again, I wanted to just tell you the power of Raman spectroscopy here. One other thing that can also be done is, as new substances are being added and are coming into the market, and as they become controlled, Raman spectroscopy has the power to identify them by getting new libraries in place. So if you can get these new fingerprints onto the device, you can basically use that to identify new substances like these bad salts. So that talks about 4 different examples that I wanted to just go through as to how it can be, how Raman spectroscopy can be used for identifying substances. I also want to get into a little bit about where it can go. I strongly believe that Raman spectroscopy has the power to redefine some of these existing practices, and sometime in the future, I think they will pretty much replace all NIC kits and become the go-to standard for identifying drugs in the field. It also enables law enforcement to basically change their methodology because you're gathering some more trend information that is useful for doing this forensic intelligence. And I think the new phenomena is going to be just doing policing based on forensic intelligence. Here is one example. If you have these devices that are out there and they're all networked together, these Raman spectrometers, you can use time, date, location, and even the mixture analysis of these cutting agents to find out where your source of these drugs are coming from. So hypothetical example would be is if, let's say, there are 3 devices, in South region, West region, and East region, and you're seeing the same exact mixture of cocaine and caffeine coming from the South region, and they start begin to happen in East and West, then then you can sort of triangulate where your source for these different drugs are coming in. And this trend information and intelligence information is very, very powerful that can be used, especially if these devices are networked together where they can share the spectra evidence amongst each other. This again, another new approach. This will take time for the systems to change. Right now, this is a good presumptive identification tool. But in future, I can also see this becoming a confirmatory science, where the field officer would just gather the spectra. Previously, what used to happen is the field officers would gather the evidence, lock it up in an evidence bag, and then send the evidence sample back to the lab and sort of mail it or FedEx it or whatever. And then there's a turnaround time for analysis, and then you will get the results back in. I could see a system where if the substance is basically identified in the field, if the system that the crime lab is also using is similar, you can just only send the spectra or email the spectra out to the lab and let the lab verify that that spectra is verified as a cocaine spectra. So you can at least cut down on the turnaround on evidence and all that kind of stuff, and all you're doing is just sending spectra back and forth in the crime lab and the field office, and you can get those confirmatory evidence. Long term, I think there is potential for doing some of the confirmatory evidence analysis by these devices in the field itself. But again, Raman spectroscopy at this time is sort of limited to presumptive identification at this point. So what is the legal perspective on this? Raman spectroscopy as a science itself has been accepted as court evidence in a lot of federal courts. There are several examples where Raman spectroscopy has been used for those in those cases. These are all at U.S. federal district court level. These are 3 examples. Most of them are pharmaceutical patent litigations about, right pharmaceutical tablet versus the tablet that's basically made by a different manufacturer. In several of these instances, Raman spectroscopy has been used to analyze those tablets and give the appropriate judgment based on that. So Raman itself has passed both the Fry standard and the Daubert standard, which are the standards that the judicial system now in several jurisdictions is using to make sure that the evidence that is basically being presented passes this muster, or the muster of scientific evidence behind it. So Raman spectroscopy has passed both the Fry and Daubert standard, and these are some 3 example cases. Individual devices would have to go through the process itself, like even for MFL 3000 itself, would have to go through the judicial process. But the science itself has been accepted in the legal world. So in summary, I would want to validate that Raman spectroscopy is an emerging approach that is becoming available in user-friendly devices, and they are a much better alternative than the existing process tools that are out there. The systems approach is a big force multiplier as well. It sort of has the CSI effect when defendants see analysis done by these devices, they immediately just basically plead out. So there is bigger plea bargains that are happening because of use of Raman spectroscopy-based devices. Of course, there are some constraints like any other technology would have, but I believe those are all very manageable constraints. And I think by endorsing and by embracing these technologies in the legal system, I think it'll enable a faster justice system is what my belief is. With that, I would like to thank you for your time. If you need any additional information on Raman spectroscopy or specifics about MFL, please feel free to reach out to us, and we'll be happy to answer any questions at this point.
Okay. The first question we have, Prasant, is how are the courts working with the MFL 3000?
MFL 3000 is accepted as a presumptive identification device in several jurisdictions. Again, it depends on the particular jurisdiction itself and what the DA feels about it. It has already been accepted in several jurisdictions right now, but strictly, again, as a presumptive identification system at this point.
Great. The next question was, can you detect counterfeit pills?
Yes, that's the beauty about MFL 3000 is it's not analyzing drugs based on markings. It's analyzing them based on the chemical analysis of the tablets. So if you have, there was one example where there was an authentic, actually a fake Cialis was presented at the device, was not able to come back and say this is Cialis because the chemical makeup of it did not match up with that. Yes, so it is good for counterfeit pills as well.
Okay. One more is what is the lifetime? How long does the unit last?
These devices, Raman spectroscopy devices, have been in the market for quite some time now, actually. So they are known to be very robust. Specifically, our device, MFL itself, has been through live testing so far, and we believe that it's good for. There are no moving parts in these devices, so it's good. 6-7 years is an average lifetime that I would want to put on these devices, but I can see them lasting longer than that.
Wow, great. And finally, are there any limitations, or could you expand a little bit on the limitations to the drugs you might be able to test?
Yeah, right. I mean, most technologies have some constraints and limitations, and Raman spectroscopy is not an exception to that. For example, Raman spectroscopy is not really good for like marijuana type plant materials because there is a lot of fluorescence coming back from them. So it won't be good for that. And it's also not exactly a trace technique. So if you have very small amounts of substances like at nanogram level, this is not really the technology for identifying substances at the nanogram level. It's mainly good for bulk powders and bulk substances. Something where your concentration of the sample is at least 10% or better.
Okay.
So those are the main limitations.
Great. If there are not any more questions, I'm not seeing any more come in right now. If you have any, just send us a quick chat. That will conclude the webinar. I just want to remind everybody that we did record it, and we will be sending the recording out in a couple days. And if you have any questions, please feel free to contact the email that's on the screen in front of you. Thank you.